Hello, welcome to Biology, the science study of life. Identifying clones carrying a gene of interest. To screen all the colonies with recombinant plasmids, the white colonies in the above method. For a colon of cells containing a gene of interest, we can look either for the gene itself or for its protein product. In the first approach, which we describe here, the DNA of the gene is detected by its ability to base pair with a complementary sequence on another nucleic acid molecule, a process called nucleic acid hybridization. The complementary molecule, a short single-stranded nucleic acid that can be either RNA or DNA, is called a nucleic acid probe. If we know at least part of the nucleotide sequence of the gene of interest, perhaps from knowing the protein it encodes or its sequence in the genome of a closely related species, we can synthesize a probe complementary to it. For example, each part of the sequence in one strand of the desired gene is like this: 5 2 3. Notice that. Then we would synthesize this probe. Notice that 3 to 5 direction. Each probe molecule which will hydrogen bond specifically to a complementary strand in the desired gene is labeled with a radioactive isotope or a fluorescent tag, or so we can track it. For example, we could transfer a few cells from each white colony in this figure. Notice that. To a spot on a new agar plate and allow each to grow into a new colony. This figure shows how a number of such bacteria colonies can be simultaneously screened for the presence of DNA complementary to a DNA probe. An essential step in this method is the denaturation of the cell's DNA, that is, the separation of its two strands. As with protein denaturation, DNA denaturation is routinely accomplished with chemicals or heat. Once we've identified the location of a colony carrying the desired gene, we can grow some cells from that colony in liquid culture in a large tank and then easily isolate large amounts of the gene. Also, we can use the cloned gene itself as a probe to identify similar or identical genes in DNA from other sources, such as DNA from other species. Storing cloned genes in DNA libraries. The cloning procedure in this figure, which starts with a mixture of fragments from the entire genome of an organism, is called a shotgun approach. No single gene is targeted for cloning. Thousands of different recombinant plasmids are produced in step 3, and a clone of each ends up a white colony in step 5. The complete set of plasmid clones, each carrying copies of a particular segment from the initial genome, is referred to as a genome library. Notice that. Scientists often obtain such libraries or even particular cloned genes from another researchers or a commercial source, sometimes referred to as cloning by phone. Certain bacteriophages are also common cloning vectors for making genomic libraries fragments of foreign DNA can be spliced into a phage genome, as into a plasmid, by using a restriction enzyme and DNA ligase. An advantage of using phages as vectors is that a phage can carry a larger DNA insert than a bacterial plasmid. The recombinant phage DNA is packaged into capsids in vitro and introduced into a bacterial cell through the normal infection process. Inside the cell, the phage DNA replicates and produces new phage partials, each carrying the foreign DNA. A genomic library made using phage is stored as a collection of phage clones. Since restriction enzymes do not recognize gene boundaries, some genes in either type of genomic library will be cut and divided up among two or more clones. Researchers can make another kind of DNA library starting with mRNA extracted from cells. The enzyme reverse transcripts obtained from retroviruses is used in vitro to make single-stranded DNA transcripts of the mRNA molecules following enzymatic degradation of the mRNA. A second DNA strand, complementary to the first, is synthesized by DNA polymers. This double-stranded DNA, called complementary DNA or short-term cDNA, is then modified by addition of restriction enzyme recognition sequences at each end. Finally, the cDNA is inserted into vector DNA in a manner similar to the insertion of genomic DNA fragments. The isolated mRNA was a mixture of all the mRNA molecules in the cells used, transcribed from many different genes. Therefore, the cDNAs that are cloned make up a library containing a collection of genes. However, a cDNA library represents only part of the genome, only the subset of genes that were transcribed into mRNA in the original cells. Genomic and cDNA libraries each have an advantage depending on what is being studied. If we want to clone a gene but are unsure in what cell type it is expressed, or are unable to obtain that cell type, a genomic library is almost certain to contain the gene, or to contain the gene. Also, if you are interested in the regulatory sequences or introns associated with a genomic library is required, because these sequences are absent from the fully processed mRNAs used in making a cDNA library. If this very reason, if you are only interested in the coding sequence of a gene, you can obtain a stripped down version of the gene from a cDNA library. A cDNA library is also useful for studying genes responsible for the specialized functions of a particular cell type, such as brain or liver cells. Finally, changes in patterns of gene expression during development can be traced by making cDNA from cells of the same type at different times in the life of an organism. Back to genomic library. Notice this figure, this one bacterial clones, and this one phage library. A genomic library is a collection of many bacterial or phage clones, each containing copies of a particular DNA segment from a foreign genome in a complete genomic library. If foreign DNA segment cover the entire genome of an organism, notice that this one represents, notice that shown are 3 of thousands of books in a plasmid library. Each book is a clone of bacterial cell, each containing copies of a particular foreign genome fragment, pink, yellow, black segment. Notice that pink, black segment, and this one. Notice that, boom. Um, Closer look. Notice that this one closer look. In its recombinant plasmid. Notice B also. The same 3 foreign genome segments are shown in 3 boxes of a phage library. Notice that. Okay. Back to nucleic acid probe hybridization. Notice also hybridization with a complementary nucleic acid probe detects a specific DNA within a mixture of DNA molecule. In this example, a collection of bacterial colonies are screened and identify those carrying a plasmid with a gene of interest. First, notice that a special filter paper is pressed against the master plate, transferring cells to the bottom side of the filter. Marks X, notice that, are placed on the filter paper and eager to establish the position of each individual colony in reference to the marks. Notice also here, notice that, boom. The filter is treated to break open the cells and denature their DNA. The resulting single-stranded DNA molecules are treated so that they stick to the filter. Radioactive probe molecules complementary to part to the gene of interest are incubated with the filter. Notice also film here and master plate here. Boom. Okay, the result, colonies of cells containing the gene of interest have been identified by nucleic acid hybridization. Cells from colons staged with the probe can be grown in large tanks of liquid growth medium. Large amount of the DNA containing the gene of interest can be isolated from these cultures by using probes with different nucleotide sequences. The collection of bacterial clones can be screened for different genes. Okay.